Good luck. I'll try to get through this. It's a little extra time for you. So, what I'm going to talk about for you is RNA viruses. And I know that RNA viruses probably aren't the highest priority for what you think you're interested in and what you're studying. But there are some really important aspects of these viruses that you should be aware of. And they turn out to be really important pathogens. And there are plenty of RNA viruses that are going to walk into your offices. And you're going to have to deal with them from your patients. So you want to be aware of that. You want to be aware of that. So for these lectures, we're going to talk about the properties of RNA viruses, the life cycle of these viruses, and we'll share a brief overview of host recognition and responses, and then we'll talk about a category of RNA viruses, emerging pathogens. That's for today's lecture. So, thinking about RNA viruses, I thought maybe you knew a few of the answers to these questions. Think about these for a moment. Can you name the virus? So this RNA virus caused an outbreak in Western Africa in 2014. And in some circumstances, it can kill over 80% of the folks who get infected with it. Without treatment, this RNA virus nearly always results in death. And it remains a problem in parts of Africa where domestic dogs are able to spread the infection. Starts with an R.
Rabies.
Good. This virus emerged in the early 80s and has resulted in the death of over 36 million people worldwide. HIV. This year, this RNA virus, or each year I should say, this RNA virus is to blame for about 500,000 people dying. Again, that's worldwide. Any guesses? How about influenza? Over 40% of the world's population is considered at risk for this virus, which can cause an Ebola-like illness. It doesn't usually cause an Ebola-like illness, but it can. And it is responsible for infections of 50 to 100 million people each year. This is a hard one. This is dengue virus. And despite the availability of 2 good vaccines, this virus caused the death of over 400,000 children under the age of 5 in 2008. Rotavirus. All right, so those are the answers. All right. So our learning objectives as we go through these RNA viruses, hopefully I just impressed upon you that they're kind of important, particularly globally. We want to understand the viral structure and the major criteria for classifying viruses into families. We want to describe the major steps in a prototypical virus life cycle, describe general mechanisms of RNA virus, the RNA viruses used to replicate, appreciate the differences between what are called positive-stranded RNA viruses and negative-stranded RNA viruses, review some general mechanisms of host defense, strategies for vaccine design, and understand the concepts of herd and ring immunity, and also understand the role of RNA viruses as emerging pathogens. So that's quite a bit, actually, to get us started. I know some of this is going to be review for you, and that's okay. Enjoy that part so you don't have to think too hard, and then concentrate on the things that you're not as familiar with, obviously. So viruses in general are small, obligate infectious particles. They contain nucleic acid, and the nucleic acid is covered by protein. These are obligate intracellular parasites, so they rely completely on the host cell in order to replicate themselves. And for a long time, people will say, well, are viruses alive? Are they not alive? They're sort of one of these things that's on the edge of life. And generally, we think about living organisms, we characterize based on their characteristics. And so, viruses share a number of properties with living organisms, including their nucleic acid genome, their capacity to replicate, make progeny, inherit genetic information, which means that they can evolve, mutate and evolve, and that's very lifelike. But some of the things that they generally lack is a basic cell structure, and they lack the cellular machinery needed for protein synthesis, like ribosomes, and they do not have the property of metabolism, so they can't make their own energy. They've got to basically steal the host cell for whatever energy they need, whatever replication they need to do in terms of protein synthesis and so on. And so many people think that viruses are not really, truly alive. We don't have to worry about which side of the debate you want to follow. What we do know is that viruses are small, as I mentioned already. They are around the size of a ribosome. And so if you look at the scale of life here, you got these cells, then bacteria. Viruses are falling in this category right here, around the size of a ribosome. And so they're like 20 to 500 nanometers in terms of their size. So very small, and you can appreciate that on this T cell here that is budding virus. These little yellow guys are HIV virions budding from the surface of a cell. So you can appreciate that these are very small little guys. And it's that property, the fact that they're so small, that led to their discovery. And this is attributed to an RNA virus. So here's something that we can talk about for RNA viruses and toot that horn. So RNA viruses, so plant virus in particular, tobacco mosaic virus, was the first virus described. And people noticed that it was able to infect plants, or something was able to infect plants and cause this mosaic sort of appearance on the leaves. And that was known for a long time. And then folks started trying to figure out, well, what's causing this? And one of the techniques that they used early on, that Dimitri used and Martinez, they started filtering extracts from these leaves and testing their ability to transfer infection. And what they found is that they used filters that typically got rid of bacteria, they still had an infection that they could pass on to another plant. And so that was sort of their clue that this might be something different. Dimitri, the Russian scientist, thought that this was probably just a really small bacterium or a toxin. So it wasn't really on top of the difference in the cause. And whereas Martinez, he coined the term virus, he thought of it more of a slime, a fluid that was conferring infections. And it wasn't until later, when folks really were able to look at viruses by electron microscopy, which was invented in the 1930s, that people really appreciated the particulate nature of viruses. The first human virus discovered was yellow fever virus. That was in 1901. So what about the structure of these viruses? Well, you're going to hear some terminology. You're probably familiar with a lot of it. One is the virion itself. This is a form of the virus that exists outside of the cell. It has at minimum a protein structure surrounding a nucleic acid. What is that protein structure? Well, it's called a capsid. And these are made of viral proteins that have the property of being able to self-assemble. So the idea is to build a relatively simple structure and use the least amount of energy and proteins as you can to make a simple structure that can protect the nucleic acid and help it get to its next host cell. So that's the role of the capsid. So the capsid plus a nucleic acid core is often referred to as a nucleocapsid. And there are also parts of the virion that include packaged enzymes in some cases, where the virus enzymes actually go along for the ride with the virion itself. And viruses will have, in some cases, an outer membrane, an envelope, that is derived from host cells. So you'll you will hear the terms naked and envelope viruses. So the envelope viruses have the envelope, naked or just protein on the outermost surface. So a closer look at the capsids. There are three basic types: the icosahedral capsids. Anyone know the generic name for the type of enzymes that this virus might want to protect from? So it's a DNA or RNA genome you're trying to protect. So nuclease, good. So the particle will help protect it from nucleases. Other viruses use this sort of helical structure, and they arrange it in this sort of circular fashion that you see here. This is a sort of a filamentous type structure that one can see. And then other viruses have more of a complex structure that don't really fit into either of these categories. The virus that causes smallpox is one example of a complex virus. So what about this naked versus envelope viruses? Or some basic concepts here. One is that the capsid in the naked virus is what protects the genome from the outside. That's the outermost component, whereas the envelope is deriving host-derived membranes. They may both, depending on the virus, have these glycoprotein spikes that extend out and are able to allow the virus to interact with host cells. So they tend to be the receptors that the virus uses to target its host cell. The naked viruses in general have a better capacity to survive in the environment. They're more stable, and they're also more stable in the GI tract. So a lot of viruses, like rotavirus, that causes infections in the GI tract, have these really complex, more layered capsids that help them survive. Envelope viruses are not as good at surviving in the environment because, of course, they have this biological membrane that if you get rid of it, now you have a real problem in terms of the ability of that particle to infect anything. But it does have an advantage in terms of potentially escaping from the immune system. It's sort of hiding most of the protein structure of the particle inside of this envelope protein that's host-derived. And so there's less for the host to see and respond to. And there's also an advantage to envelope viruses in terms of their release from cells. They can bud from the surface of cells, and often they can be released in a way that does not lead to the death of the cell or the rapid death of the cell. And this can promote their ability to be propagated longer in an infected host cell. These types of viruses use different ways of getting into the cell. The naked viruses often use pore-forming mechanisms to penetrate through membranes. And the envelope viruses use fusion proteins that allow them to fuse with host membranes. So viral genomes, we have the DNA viruses and the RNA viruses. The DNA viruses in general have larger, more complex genomes. There are some DNA viruses that have really small genomes, but in general, they're the more complex and the larger ones compared to the RNA viruses. The RNA viruses, in terms of their genome, they can be single-stranded, double-stranded, linear, segmented, positive, and negative stranded. And we'll talk momentarily about what that latter part means. The RNA viruses, most of them will replicate in the cytoplasm. Most. The enzyme that catalyzes their replication is called the RNA-dependent RNA polymerase. It is prone to errors during replication, and so RNA viruses in general have a higher mutation rate than DNA viruses. And this is thought to contribute to their ability to adapt to new species and to contribute to these emerging diseases that we often see. So the RNA virus genome, again, single-stranded or double-stranded, and positive-stranded or negative-stranded. So what does that mean? The positive strand of an RNA virus is basically messenger RNA. So it can be translated into protein. It can be directly translated into protein. So a positive-stranded RNA virus, once it gets into the host cell, it's ready to make some proteins right from the genome. And so it can get things moving very quickly. The RNA, the negative stranded RNA viruses will have to make a positive strand, will have to make messenger RNA from their template in order to make protein in the host cell. So these are the two key differences that you want to be familiar with in terms of the RNA virus genome, what the positive strand means versus the negative strand. And of course, some of them are segmented. This refers to genes on the RNA which appear in segments. So there may be 3 segments, there may be 9 segments, just depending on what virus we're talking about. Not all viruses have segmented genomes, just some do. And each segment generally encodes for one protein, and that's why it's called monocistronic RNA. Whereas non-segmented genomes tend to produce polycistronic mRNAs or a polyprotein that can be cleaved into multiple proteins. Okay, so with these this basic understanding of what makes up a virus and how it's structured and so on, people like to organize viruses into families and classify them and try to group them into which viruses are more like other viruses and so on. And so there are 3 major criteria that people use to do this. One is the nucleic acid itself. Is it a positive stranded, negative stranded? Is it a double stranded RNA virus? So the genome makes a big difference. The capsid structure. Is it icosahedral? Is it helical? Is it complex? So these get further divide. And then, is the virus enveloped or naked? And these are sort of the big three, if you will, for how folks classify viruses. And there are, orders, families, and so on, but what you practically see when you're reading about viruses are the families, which end in the suffix viridae, versus the genus and the species, which both end in virus. So that just comes from experience in terms of understanding whether you're talking about a species or a genus. It's unfortunate, but that's the way it goes. So this is sort of the typical scheme of RNA virus classification. And so you can see that you start with these RNA viruses and you ask, well, is it single-stranded? Is it double-stranded? Is it positive sense? Is it negative sense? And then you go from there and you say, well, is it enveloped or is it naked? And after that, you can group viral families into different categories. And further subdivisions are going to be based on other criteria about, what does the virus do to the person in terms of the infection, what's the relatedness of the proteins, as molecular biology has moved forward over the years. So these different things are used to further classify them. But these are the big 3 that people think about. A different type of classification system comes from David Baltimore, and this is more recent. And Baltimore described viruses based on how they replicate, and how they use their strandedness to make messenger RNA, basically. And so he's classified them into 7 groups, and we only have to worry about 4 of them in this class, which are the RNA viruses. So group 3, the double-stranded RNA viruses. Group 4, the positive sense single-stranded RNA viruses. Those things include the pornoviruses, togoviruses, so things like your cold virus, typical cold virus, or poliovirus. Negative sense stranded RNA virus, so the orthomyxoviridae, as an example, that's the influenza virus. And then, of course, reverse transcribing genomes that start with RNA, but then make DNA from the RNA. Okay, these are retroviruses. And HIV, of course, is our prototypical example of a retrovirus that causes problems in humans. So if you take a closer look at the Baltimore classification, it's all centered around messenger RNA. How does the virus make messenger RNA? And again, focusing on the RNA viruses, which are shown in green here, the group 3 viruses, which are double-stranded, act a lot like the negative-stranded viruses, which are group 5. And those viruses will use their negative RNA template to make positive stranded RNA that serves as messenger RNA. And then they can make proteins and go on from there. The negative or the positive strand RNA virus can serve as a messenger RNA in itself. The genome is ready to go. It's ready to be translated, gets into the cell, starts making protein immediately, and so that virus can serve as its own message. Alternatively, the positive stranded RNA can also make a negative stranded template, which can then make a positive stranded RNA or another message. So it has two ways to make messenger RNA. One is just from the genome itself, and the other is to make that template of negative stranded RNA virus and then make more positive stranded RNA viruses. And then the last group are the retroviruses, which use an enzyme called reverse transcriptase to use that RNA genome to make a DNA copy that then goes into the nucleus, gets inserted into the nucleus, and from there you can make message just like you would from any old DNA that's in the host. So these are the different ways of making messenger RNA. That's what the Baltimore system is all about.
Question.
Yeah.
What benefit would they have if it's already positive stranded RNA? Why would they make a negative strand to then go back and make another positive strand? Wouldn't it be the same they were starting?
They make the, one of the important things about that process is that the positive stranded RNA viruses, they make the negative strand, but then that is how they amplify their genome. So they make the negative strand and then they make multiple copies of the positive strand. And what you end up with is a lot more positive stranded RNA than negative stranded in those particular viruses. So it's a mechanism to enhance their capacity to replicate their genome. So these viruses then, if you take a look at the key enzymes in these viruses, there's two key enzymes. One is the RNA-dependent RNA polymerase. Okay, so that means it's RNA dependent, it uses an RNA template, and it's an RNA polymerase, so it's going to make a new strand. So it's RNA dependent RNA polymerase. And the three types of viruses that use this method include the Baltimore's groups 3, 4, and 5. And so these are your negative, positive stranded RNA viruses, double stranded RNA viruses. The retroviruses use a different method and a different enzyme, which is the reverse transcriptase enzyme. So that's the key enzyme for the retroviruses, whereas the RNA-dependent RNA polymerase is for the other virus groups. So what about this RNA-dependent RNA polymerase? Well, some of these viruses actually need it in order to get themselves started. And so they will package RNA-dependent RNA polymerase within the virion itself. And so your negative stranded RNA viruses and your double stranded RNA viruses that need to make a positive strand RNA virus in order to get protein translation going, they're going to need that RNA-dependent RNA polymerase in the virion itself. So this is one of the differences between these double-stranded and negative stranded RNA viruses and the positive stranded RNA virus. The positive strand doesn't really need the RNA-dependent RNA polymerase in the virion because it can make that protein once it infects the cell. And it can make the message that it needs to, or it is the message that it needs to make the protein. And so that's how they are divided. And of course, the retroviruses use our reverse transcriptase, and so they use a different enzyme altogether. So let's think about this momentarily. Covered a few things already. RNA viruses tend to have more or less complex genomes than DNA viruses. Less. Good. Which of the following types of RNA genomes can serve as a direct template for protein translation? Positive stranded RNA viruses, right? Which of the following is not considered one of the three major characteristics used to classify viruses? The genome sequence, the capsid structure, the strandedness, is it positive or negative, or the presence or absence of an envelope?
Genome sequence.
Genome sequence, right. So the big three: capsid structure, strandedness and envelope or not. All right, so those are some just basic concepts that we've already covered. Okay, what about the virus life cycle? This is sort of a generic view of a virus life cycle. And so the virus has to accomplish several things, and you're probably already familiar with this because you've talked about DNA viruses, I think, already, so you get the idea already. But they have to enter the cell, and so there has to be attachment and penetration. And then once they enter the cell, they uncoat, they release their genome from the capsid. Okay, so in this figure, I'm showing you an example of a virus that binds to some receptors. So those glycoproteins on this particular virus, and this represents an envelope virus. So the outer part of this virus is an envelope, and it has its little glycoproteins, and they're binding to a specific receptor. And through that interaction, that virus the membrane is fusing with the host membrane. In this case, it's fusing at the cell surface. And it's letting its capsid with the genome into the cell. So this is, HIV gets into cells this way. Parainfluenza viruses get into cells this way. They interact with the cell membrane and get in. Other viruses we'll see are endocytosed into the cell. And then in the endosome of the cell, they interact and are able to fuse and get into the cell that way. So once the capsid is somehow gotten into the cytoplasm, it releases its RNA genome. And this can be sometimes a very structured and carefully delivered mechanism. So for example, HIV, the capsid comes in and it interacts with some host proteins, and it actually hijacks the machinery of the cell to the microtubule machinery, the cytoskeleton, to travel to the nucleus. And once it gets there, it uses some other host proteins to actually get itself uncoated and get into the nucleus itself. And so this can be very carefully orchestrated in some circumstances. So the uncoating releases the genome from the capsid, and then the virus will go through its genome replication and its protein synthesis while it's in the cell. And so with the RNA viruses, it makes these proteins. The proteins then assemble into capsids, and they package the genome, and then they find a way to get out of the cell. And this can be through a budding process, which is very common in the envelope viruses, or sometimes the cell itself will just die through lysis of naked viruses that gather in the cytoplasm, and with their pore-forming molecules are able to punch holes in the membranes and ultimately escape from the cell. All right, so let's break this down just a little bit more. Entry, most of the time we're talking about a very specific interaction between a receptor and a ligand. So this determines tropism of the virus. Tropism, what cells is it going to infect? Okay, and this is really important for determining pathogenesis, as you might guess. An example is HIV. Infects CD4 T cells, you have a severe immunodeficiency. Okay, influenza infects cells in the respiratory tract, you get a severe respiratory infection. And rotavirus infects cells in the intestine, you get a severe intestinal infection. So tropism is very much related to the pathology of the virus. Now, once the virus is bound to the cell membrane, it has to enter the cell, and it can either do this, as I mentioned already, by getting through the plasma membrane or getting through the endosomal membrane if it's endocytosed. So envelope viruses tend to get in through fusion. Okay, so they utilize special fusion proteins that are part of their glycoprotein makeup, and that helps them get into the host cell. And naked viruses tend to form pores in plasma membranes, oftentimes within the endosome, or causing endosomal disruption, which basically breaks apart the endosome, releasing the virion into the cytoplasm. Viruses will then uncoat, the capsid is removed, and the genome is exposed, and this, of course, is critical in order to start replicating proteins, in order to start replicating the virus itself. And there's nothing really special about this comparing RNA and DNA viruses. So what about gene expression and replication strategies? A couple basic concepts to keep in mind. One is that most of the RNA viruses are going to replicate in the cytoplasm. A well-known exception to this rule is influenza virus. It's an orthomyxovirus that replicates actually in the nucleus. It's into the nucleus and replicates its genome from there. There are also, among RNA viruses, high mutation rates, which I already mentioned. So these could be like 10 to 100-fold greater than a DNA virus. And this contributes to their capacity to move around, to evolve, and infect different species. And another big player in the diversity of viruses that spring from these RNA viruses come from these segmented viruses, like influenza, which can actually, you can have two viruses infect a cell or infect a host, and they can actually swap some of their segments. And so you can get a virus that's circulating in a bird, and or a wild bird, and it gets into a domestic bird, and they swap some of their RNA genome. And now you have a new virus that has different properties. And so this is one of the ways that creates diversity in those types of viruses. So that's called reassortment. Now RNA viruses have a problem. They don't necessarily have the perfect structure of a typical messenger RNA molecule that's found in the host cell. And so if you look at host cells and most of their messenger RNA, you'll find what are called cap structures. Okay? And these are these methylated 5' ends of the messenger RNA that have a 7-methylguanosine attached to them. And this turns out to be really important for the messenger RNA and its interaction with ribosomes and its ability then to actually translate proteins. Otherwise, it's kind of floating around and it can't finish its job, even though it has all the message it needs, the whole template. It can't interact with the machinery of translation. So viruses then have to come up with different ways of dealing with this. And so some of the ways that they do this, some of them can, of course, just use the host machinery and make caps from the host machinery. And so this is a property of retroviruses. And if you recall, retroviruses start out as RNA. They change into their genome in the DNA. The DNA inserts into the genome of the host cell. And from there, it can be transcriptionally controlled, and translation, and the message from those transcripts can be translated. And as they're being produced, host proteins or host enzymes can help form their cap structure. So some viruses can actually use the host machinery for their own purposes and make a cap that's very much like the cap of any message. But some viruses have to steal the cap structure. So influenza, for example, is a cap stealer. And it has enzymes that will cut off the cap of a host messenger RNA molecule and hook it on to their positive stranded RNA. And so that then becomes a competent message that can interact with host proteins and can then translate new proteins for the virus. So that's called cap snatching. And then some viruses will encode their own capping machinery, and others don't use a cap at all, but they use proteins that are linked to the ends of the virus, or they'll have these special structural components of the RNA genome that sort of take the place of the cap. So this is an example of what's called non-template functions of viral RNA genomes. And shown here is this really wild, diverse structure on the 5' end of this. This is poliovirus. And it has this VPG protein at the very end, and it's attached to this cloverleaf-like structure. And then you have this iris thing of the internal ribosomal entry site. And so you have these sort of complex RNA structures that don't encode for proteins, but they do have an effect on viral translation and replication. And so in this case, the iris, for example, this section of the RNA sort of takes the place of a cap structure, and it can interact with ribosomes. And so it doesn't need a traditional cap in this case. It has this other complex structure that allows it to mediate its functions. And then cloverleaf structures can also, they interact with proteins, host proteins as well as viral proteins, and this can also facilitate either translation or transcription of the viral template. So non-template functions of RNA genomes can be important, particularly in these RNA viruses. And then if you break down the RNA viruses into the 3 categories of positive, negative, and double-stranded, and we're leaving the retroviruses out of this for the moment, there are some sort of general basic properties that these viruses have. So the positive-stranded RNA viruses undergo what's called asymmetric replication, and that is what we were just talking about a moment ago. The positive strand can be used to make protein, but it's also used as a template to make RNA, negative-stranded RNA, and then the negative-stranded RNA becomes a really important template for making more positive-stranded RNA to make more protein, and also for making genomes that will then serve to infect new cells. So it turns out that you end up with a lot more positive-stranded RNA than negative-stranded RNA in those particular viruses. They also tend to utilize host membranes to a rather extensive amount. And so you'll find these viral proteins anchored into various host membranes like endoplasmic reticulum. And this is where the viruses mediate their translational properties, their transcriptional properties. And so we'll see one example is the hepatitis C virus, which is a very complex life cycle that utilizes these sort of membranes to get its work done. The negative-stranded RNA viruses have these ribonucleoprotein complexes. So there are proteins that sort of associate directly with the genome itself. And they tend to have these helical shapes, and the proteins then help the function of the virus in terms of viral RNA replication and transcription. So those play an important role. The double-stranded RNA viruses, these are viruses that have frequently use what's called subviral particles or replication in subviral particles. So the capsid doesn't come apart fully when it gets into the cell, and it has these little, it's able to mediate most of its replication actually in the capsid itself, and then likely the viral transcripts that are produced exit through pores that the virus is able to generate. So those are some basic differences in the RNA viruses. And finally, assembly and release. Again, this one isn't very different. So you have new viral genomes and proteins that are assembled into new virions. The viral capsid is frequently partially assembled in some spot, and then the genome kind of hooks into it. And there are proteins that help facilitate that interaction. And then the nucleocapsid can undergo further maturation as the virus matures completely. There's some evidence, for example, in HIV disease, that the new virus buds from the cell surface, that all the proteolytic processing of the virion isn't really complete. And it kind of happens very rapidly after release. And there's some debate about that, but the virus may not always come out fully ready to infect host. Non-envelope viruses, these newly formed virions accumulate in the cell, and then they can be released by lysosing the cell. Whereas we already mentioned the budding part of the envelope viruses. And keep in mind that envelope viruses can bud from various types of host cell membranes. So, a virus fusion event that likely results in the entry of the virus at the plasma membrane of the cell is most likely pH dependent or pH independent? You have to think about this one. It's happening at the cell membrane. Maybe to help you think about where else does the virus get in? The endosome, right. And what would you predict in the endosome? What kind of environment is in an endosome? Acidic, right? So as an endosome matures, it becomes more and more acidic. Hydrogen ions move their way into the endosomes. And that acidic microenvironment then can induce conformational changes in the virus that allows it then to fuse or mediate its pore-forming activity with the endosomal membrane. Okay, so in that case, that's a pH-dependent event. Whereas at the cell membrane, the pH is not acidic, and so that would be an independent event. So that was a harder one, that was something you have to think about a little bit. A virus that belongs to the Baltimore group 3 generates viral transcripts while partially associated with a capsid as a genome that is group 3. Double-stranded RNA, very good, very good. All right, let's talk about immune protection a little bit. Take a little break. Unless you guys want more time at the end to study, you can keep going. Try to get out a little early. That's perfect. All right. So let's talk about immune protection and viral infections. This again, this is probably review for you. Already had some immunology, and so this is just sort of a refresher to highlight some of the most important mechanisms of the host in terms of fending off these viruses. And so one concept that we have to appreciate, there's this innate defense system and there's the adaptive immune system. An innate defense is a rapid responder, if you will. It has cells that are ready to respond to any challenge. It may also just constitute barriers and antimicrobial peptides and other things that are already present and already prepared to respond to any incoming insult. Whereas the adaptive immune system has the property of memory, can respond to something that the host has seen before in a more rapid and robust way, and has what's called specificity associated with it. So it has these special receptors that allow it to clonally expand cells that respond to specific proteins that might be associated with the viral pathogen or other pathogens. But so let's think about the innate defense momentarily. These guys, this is important for defense against viruses. And one of the first things that has to happen when a virus enters a host is it has to be recognized as trouble. And so there are these recognition or innate receptors that are specialized to respond to components of viruses or bacteria or other pathogens, and they induce certain types of immune responses. And so viruses, one of the big differences about viruses and host cells is the genome, the nucleic acids that are associated with the viruses. And so the host cell has these receptors that allow it to recognize things like uncapped RNA or double-stranded RNA or double-stranded DNA. And some of the things you may have heard of are RIG1, TLR3, TLR7. And they can also recognize certain glycoproteins like TLR2 can do that. But mostly when we think about viruses, we think more about the nucleic acids being recognized. That's not a strict, that's not always the case, but keep that in mind. Now, one of the things that's really important in innate defenses against viruses are these type 1 interferons and type 3 interferons. So type 1 interferons, does anybody know the names of those guys? Remember type 1 interferons? All right, those are antibodies. The interferon molecules. All right, well, these guys, the type 1 interferons, maybe you've heard of interferon alpha or beta. Those are type 1 interferons. Maybe you've heard of interferon gamma. That's a type 2 interferon. And I don't know if you've heard of interferon lambda or not, but that's a type 3 interferon. The interferons were named because they are able to interfere with viruses. And the type 1 interferons and 3 interferons are especially important in viral infections. So what they do is the type 1 interferons are soluble molecules. The cell gets infected. Its proteins, its innate receptors can get activated. It starts making these type 1 interferons, and they go out and they condition the cells around them to be more resistant to the virus. The type 3 interferons do something very similar, but it turns out that the receptors for type 3 interferons are more restricted. And so they're more focused on epithelial cells, for example. And so cells in the respiratory tract will make type 3 interferons and respond to type 3 interferons. The cells of the immune system will respond predominantly to the type 1 interference, but not to type 3 interference, or not as well to the type 3 interference. This is sort of the dogma, the paradigm that people think of when they think of these interferons. There are other things like natural killer cells that can respond rapidly to an infected cell, kill the host cell. And the idea is, let's kill that cell before it can make babies, make viral babies. And this is protective then to the host. So we'll sacrifice a few cells in order to save the organism. So these are just some examples. I don't expect you to know all these RIGIs and all this stuff, but just appreciate the complexity of the response that goes on here. And so you have RNA molecules, for example, short RNAs or double-stranded RNAs. They get into the host cell and they can interact with these various receptors like RIG1, which is associated with the mitochondria. And this then can lead to activation of interferon response factors, IRFs, so IRF3 or 7, and NF-kappa B activation. These are transcription factors that regulate inflammation. And so they trigger then, these transcription factors trigger then activation of host genes that are important for defense. And interferon beta happens to be one of them. Interferon beta then is released from those cells. It comes back and it interacts with its receptor. It may interact with the same cell. It may interact with the neighboring cell. And then there's this STAT phosphorylation. And this leads then to activation of STAT-dependent proteins. And so you get what are called ISGs, or interferon-stimulated genes. And the interferon-stimulated genes then go on to perform a whole bunch of different functions. There are like 300 of these gene products induced by type 1 interferons. And many of them serve some role in antiviral defenses. And of course, we don't understand all of them and all of their functions. But some of them are known. And they can inhibit virus by different mechanisms. So we don't have to go through all of those, but just appreciate that there's this repertoire of proteins that are induced by these interferons. And this is really important in host defense. So there's also the adaptive immune system, which has specificity and memory that I already mentioned, and you have these CD8 T cells, which are able to kill virus-infected cells. It's one of their claims to fame. So maybe you've heard of the cytotoxic T lymphocytes that have molecules in them that allow them to kill other cells. Does anybody know any of those molecules that cytotoxic T cell has that it's able to kill a virus-infected cell with? One of them punches holes in the cell membrane. And another one causes apoptosis of the cell. All right, well, you don't have to know these, but perforin is the perforin punches holes in target cells that are infected with viruses. And granzymes then cause apoptosis of the infected cell. And so these CD8 T cells play a really important role in their cytolytic function. And T cells in general can make a lot of cytokines, both the CD8 T cells and the CD4 T cells. The CD4 T cells are known as helper cells, and they make a lot of different cytokines, and then these can help protect, they can orchestrate immune responses of B cells that make antibodies, and through the interaction of these various cell types, the host can be protected. And oftentimes, one exposure to a virus can result in protection from re-challenge. And it's thought that the adaptive immune system is really key for that protection. And that is the basis of a vaccine. And it turns out that it's these soluble molecules, these antibodies made by B cells, that protect us in general from re-challenge with viruses and other. So they're sort of the basis of vaccines. So when you go in, you get a vaccine, the doctor wants to know, what was your titer, right, from your vaccine? Did you have a good enough immune response that your antibodies are going to be at high enough levels to protect you from infection? All right, and not surprisingly, viruses employ diverse mechanisms to interfere with host defenses, and not surprisingly, lots of viruses have proteins that target those type 1 interferon pathways. And examples include the dengue virus that we talked about already. Influenza virus is well known for this activity. And so there are different ways that viruses escape immunity. Another way is through mutation. HIV is a classic example. A person gets infected with HIV, they mount a CTL response, a CD8 T cell response, and soon enough, the virus gets suppressed, but then it mutates around it. And so you get these escape mutants, and now the virus can persist and continue to replicate. And so that can cause a lot of problems. What are the vaccine strategies that are typically used right now? And these are going to be changing, I think, in the near future. There's a lot of experimental vaccines that are using sort of different strategies, but these are what you see in commercial vaccines these days. One is the live attenuated vaccine. So the idea is to take a virus and put it in an environment where it mutates, where it adapts to a cell line in vitro or eggs, and it becomes attenuated, and it doesn't cause disease in the host. So once you put it into a person, it doesn't replicate so well because it's adapted, it's mutated and changed so it can replicate in the environment that you put it in, but it doesn't replicate as well in the human body. And so these viruses tend to be very effective at inducing T cell responses, and they can induce long-lasting immunity, and that's a big advantage of the attenuated viruses. So poliovirus, measles, rotavirus, yellow fever virus, which is probably the best vaccine that's known these days in terms of its potency, and so on. There's a lot of viruses that rely on these live attenuated vaccines. So I think the thing you want to keep in mind is sort of what's the advantage of this type of viral vaccine strategy. Then there's the inactivated viral vaccines. So an example, again, are polioviruses that have formalin-fixed virions that are used in vaccines. If you can inactivate the virus, it cannot replicate. And so it's thought to be a safer strategy than a live attenuated virus, which in rare occasions can revert back and actually replicate in the host and cause problems. And sometimes there's limitations too as to, well, can you give it to an older person? Can you give it to someone who's immunocompromised? So there are some limitations to the live attenuated approach versus the inactivated particles. There's also what are called virus-like particles, and this is based on the properties of capsid proteins that can self-assemble, and those particles can be made in vitro, basically, or in a tissue culture well, and then they can be used as a vaccine. You can induce antibodies to them, and they turn out to be effective in some cases. So, so they can be another way, another safe way to induce immunity. There's no viral genome, so there's no threat to the host. And then subunit vaccines, which rely specifically on proteins. Hepatitis B vaccine is a classic example that you need multiple injections. It's not quite as strong as some of the other vaccines, but it can be effective in some circumstances. And developing are things like DNA vaccines, where you can actually inject DNA in the host using plasmids that can replicate then in muscle tissues or other tissues and cause the production of a viral protein. And people are working on this sort of approach as well. Okay, so those are the vaccine strategies. Now, how do we apply them in the field at the population level? And so there are two things that we think about when we think about vaccines. One is herd immunity. And the concept of herd immunity is outlined in this schematic. So what you have here are blue people who are not immunized, but they're healthy. So they're not protected, right? They're susceptible to infection, but they're walking around feeling pretty good. The yellow guys are immunized and healthy, so they should be resistant to the virus that might jump into a population. And the red guys are not immunized, and they're sick. So they're bringing the virus to their neighbors. And so what you see in this schematic are different possibilities. So in the top panel, you see that the red guys, the infected folks, are going out, they're spreading their virus around to their neighbors, and because there's no immunization, it spreads like wildfire. And so the population is really in trouble. In the second group, you have some folks who are resistant, but not a lot. Some people have been immunized, but not a lot of the population. And so you still have a lot of spread and a lot of problems. And in the third example, which is what we're aiming for clinically, is to try to get enough people immunized so that when someone comes around who's infected, that will not spread as much. And it turns out that because you're protecting people who are interacting with this guy, that you're also protecting a person who might not have the vaccine because it needed to spread to them through other hosts. And so this is what you refer to as herd immunity, where if you get enough people immunized, then you can protect people who are more susceptible to the disease. So when we talk about herd immunity, one of the things people talk about is the basic reproduction number. This represents how many people in an unprotected population one infected person could pass the virus to. Okay, and you look at the numbers, mumps on the low end, 4 to 7 people on average will be infected by a person if there's no protection. Measles spreads crazy, right? 12 to 18 people from one person. And so based on these numbers and some math that I don't understand, the clinicians come up with this idea, mathematicians come up with how many people in the population do we have to immunize in order to get this herd immunity effect. And so if a virus can infect a lot of people, like measles, then we really need to get up to 95% of immunizations. Whereas mumps, you may not need to get quite that high because it doesn't spread as This is just another example of transmission of a virus that has an R value of 4. And so this means that one person can infect 4 others, but then those 4 others can infect 4 more, right? And so you get this exponential spread, and you can see how quickly something can happen. If you have sufficient immunization in the population, maybe that one person infects 1 person instead of 4, and then you can spread a little more, but eventually it kind of peters out when you've protected most of the population. So if a greater number are immune, then incidence will decline. That's really the bottom line. It's a herd immunity. So there are a lot of things that go into herd immunity. The efficacy of the vaccine, how well does it work? If you have a vaccine that's not that good, then you're going to have trouble even though you're vaccinating people. How many people are actually getting vaccinated? You know, as clinicians, you should be encouraging your patients to go out and get vaccinated, get their children vaccinated, and that's going to protect people. The idea that vaccines are bad for people has been disproven, so you should be aware of that as well. So a highly efficacious vaccine may not be sufficient if the vaccine rate is too low. And then there are other complexities that are more difficult to predict, like viral mutations. Does the effectiveness of the vaccine wane over time in a person who's been immunized as they get older? And other things like natural resistance. There are some people who are naturally resistant to viruses, like folks who have a mutation in the co-receptor for HIV seem to resist HIV. About 1% of the European population have this gene that they carry around with them. So they're very fortunate. The other concept of it, yes?
Wasn't there recently somebody who received a bone marrow transplant unknowingly from somebody with that?
Yes.
And they cured their HIV or something?
Yes.
Like that?
It's a fascinating story. I'll tell you about it next time we meet.
Sure.
But yes, it's really cool. It's not just getting a bone marrow transplant, but getting a specific type of bone marrow transplant. That was key to that success. So we'll save that for next class. But ring immunization is the next topic. And the idea here is, all right, let's not necessarily try to, sometimes it's really hard to get the vaccine out to everybody and to cover as many people as we can. And economically, it might be more wise to just try to identify cases and then immunize the people around the case. Okay, this is what they call ring immunization. So if you got the case here, the guy in red, and you immunize all the people that that person came in contact with, then you can prevent that virus from spreading. And so, and of course, isolating that person from the rest of the population is part of that strategy. So, with the ring immunization, this has been used very successfully in some cases. Ebola virus, it was actually used for smallpox, which was incredibly successful, obviously. And the idea is to identify those initial cases and jump on it right away. You have to have a good infrastructure of care to prevent that virus from spreading in these cases. All right, any questions about those topics? All right, let's see, what time we got here? 2:05, oh, we're doing good. All right, are linked to RNA viruses. So using the words of Tony Fauci and others, what are newly emerging infectious diseases? These are diseases that are recognized in the human host for the first time. So HIV in the 80s came out and was recognized, was isolated, was described. At that time, that was a newly emerging disease. Now people don't really think of it too much as an emerging disease. It's something we've been dealing with for many years. Re-emerging infections also occur, though, and these are diseases that have historically infected humans but continue to appear in new locations or in drug-resistant forms or reappear after control or elimination. So it's something that, yeah, we've known about it, it's been around, but now it's causing problems in a new location or it's under control, but now it's not under control so well, and it's spreading in a bad way. And so, you know, those are the things that we think about when we think about these emerging and re-emerging infections. And just recently in 2015, there was a meeting to describe, our group of scientists got together and said, what viruses do we really need to think about moving forward as potential emerging pathogens? And they came up with this list. A lot of them are kind of wild viruses that we probably don't have to worry too much about because they're not things that we have in the United States for the most part. But one thing they have in common is that they're all RNA viruses, every one of these guys. And so the RNA viruses are big players. So what contributes to the emergence of a disease? And there are many factors that can play into this. One is mutation, as I already mentioned, that can enhance the capacity to avoid immunity or to adapt to a new host. Climate and changes based on climate change. Animal reservoirs that might move into new geographic regions, or mosquito populations that might move into new geographic regions because the temperature is warming up in some places. These are things that can obviously affect the emergence of new diseases. Contact with animals, deforestation, again, these are things that can result in organisms getting out. Healthcare, nutrition, what's the infrastructure like? How well can we respond to emerging diseases? It turns out we can do it much better here than some places outside of the United States, which don't have the infrastructure. And that's part of their problem, and that's part of the Ebola problem traditionally, is that they haven't had the best infrastructure, although our investments have helped to change that. Travel is another issue. So over 2 billion people are traveling each year on airplanes. How many of you know the two-row rule? The two-row rule estimates of how susceptible you are. You better scratch that from the tape, please. How susceptible you are to an infection if someone is sitting 2 rows away from you and they have influenza virus, for example. So you're at a 6% risk of infection if you're within 2 rows, and it goes down to 2% if you're at least 2 rows away from them in an airplane. So this is, I don't know how important this is, but it's kind of funny stuff that people think about this in such a detailed manner. But of course, travel is a big issue, and viruses can spread that way. So here's an example of an outbreak in our own backyard, and it's hepatitis A virus. So hepatitis A is a coronaviridae virus. Okay, so pico meaning small, so a small-sized naked virus that is transmitted by the fecal-oral route. And it has a 2-week incubation, and it's a positive stranded RNA virus, which makes it group 4. It can cause liver inflammation, fatigue, jaundice, and about 1 in 5 people end up in the hospital because it can get severe, but it doesn't usually kill people, and it doesn't last long. It does last long, but it doesn't last forever. Like some of the hepatitis viruses, if you get infected, you're infected for life, like hepatitis C or B. But the outbreaks can be pretty serious, and here in our own backyard we're seeing evidence of hepatitis in different counties in Ohio, and you just get an idea. A typical year is 40 cases. We're up to 1,438 cases from 2018 through 2019. So that's a lot. That's a lot of new cases. That's reemergence, if you will, of a virus that is infecting our own state. And people have linked this to the rise in opioid use. And so, the sharing of the needles and other practices that folks do result in the spread of this virus. And this is causing major problems. So as clinicians, you're going to have to think about that as you prescribe painkillers to your patients as well. If we look at, so I listed 3 viruses to sort of run through some examples of emerging and viruses through with some that have more of a historical bend, some are a little more recent. One is the hantavirus that emerged in the U.S. in 1993, West Nile virus that emerged in Ohio in 2002, and then the Zika virus, which emerged in the Americas in 2015. So first, the hantavirus. Hantaviruses were named after the Hantan River in South Korea, and where they were originally described. These are segmented negative-stranded RNA viruses. And they have 3 segments, and each segment makes a protein. And so they're pretty simple in terms of their basic structure. But they can cause a lot of problems. There are 2 major types of illnesses that these viruses can cause. One is hemorrhagic fever with renal syndrome. That's not what we see in the United States or what we saw in 1993 when it emerged. We saw a pulmonary syndrome, in which case the person presented with things that you might think of as flu or some other more common illness like fever and headaches and muscle aches, but then they ended up with lungs filled with fluid, and it was causing a death rate of around 38%. The outbreak in the U.S. in 1993, the first case was a young Navajo Native American male, experienced shortness of breath, went to the hospital and died, and then the man's fiance died shortly after showing very similar symptoms. And this was in the Four Corners region of the United States, shown on the map there. The tissue samples from the folks pointed to a hantavirus. People had already known about these viruses, and so they found some evidence that it was hantavirus. And then they went out to their farm location, and they were able to isolate hantavirus from rodents that were hanging out in barns and around the facility. And so it turns out that the rodents don't get sick. These white deer mice that get infected with hantavirus, they spread the virus through their feces and urine. It gets aerosolized when people are sweeping things up, gets into the respiratory tract, and then can cause serious illness. And it can potentially spread to other animals as well. So why did it emerge in 93? People think, well, one of the things that contributed to it was the very rainy season that allowed for these mice to expand dramatically in terms of their population, and this may have set the stage, if you will, for the outbreaks that ended up in the human population at that time. It's not a huge number of people who are getting infected with hantavirus, so I don't think you have to stay up at night worrying about it. But it can be fatal, and we haven't really done much to improve that, unfortunately. We could do better with those numbers. Another emerging pathogen that knocked on our back door in 2002 was West Nile virus. And this is from the Flaviviridae family, flavi meaning yellow, so these viruses can cause yellowing of the skin, jaundice. Positive single-stranded genome, West Nile actually doesn't do that very much as other flaviviruses, but it has enough other similarities to the other flaviviruses that it's included in the family. Anyway, it's a positive single-stranded genome, and it's a small envelope virus. It has 10 proteins that it makes. So it has these capsid proteins and this PRM protein that's important for viral entry, glycoproteins that help the virus interact with host receptors, and a bunch of enzymes that allow it to mediate different effects in the host cell. Most people do not care when they're infected with West Nile virus. It just doesn't affect you. 20% of people end up with a fever or a rash or a headache, which isn't exactly too traumatizing. Less than 1% end up with central nervous system problems, and those can be life-threatening and major problems. The West Nile virus, its primary host is birds, where infection can be fatal. I remember when this happened, and all of the crows in my neighborhood just went, the whole population went away. It was wild. But they were really messing with my garbage cans, so I wasn't too upset at the time. So the West Nile virus infects birds. It's transmitted by mosquitoes and potentially ticks. And if you look back in history, it was around in the West Nile district of northern Uganda in the 1930s. In the 50s, there were outbreaks in Egypt and Israel. In the 90s, there were some outbreaks in Italy and Russia. And then in 1999, the first reported case occurred in New York City. And it spread like crazy through the next few years. So this is 1999 on the far left. You see that dark spot? That's where the virus started in New York. And then it started spreading. So these are like low numbers are in yellow, or wait, yellow. Yeah, I think that was the case. Darker color means more infection. And then you can see what happened in the following year in 2001. It was already in Ohio. In 2002, people were talking about it as a major concern all across the United States, essentially. So in Northeast Ohio, if you look at what happened with West Nile virus, it's kind of representative of things that happen with viral infections in general. And that is, if you look at how many people got sick versus how many people got infected. So in the case of West Nile virus, a lot of people, the ages are on the bottom, 5 to 17, 18 to 64, greater than 65. The 5 to 17-year-olds, a lot of them were getting infected. They were showing signs of antibody titers, that they'd been exposed to West Nile virus. But they weren't showing much bad disease. So the line going up is showing the neuroinvasive disease of West Nile virus. And you can see that in the folks who are greater than 65, even though fewer of them were getting infected, because they're not out playing in the yard or whatever, right? But they are getting sick. So they're more susceptible, even though they're not as frequently infected. And why is that? That there's more severe infection in older folks? Well, there are a lot of thoughts about that. One is the reduced innate immune mechanisms in older individuals, like the reduced TLR7, that is one of those innate receptors that recognizes RNA and triggers type 1 interferon production. That kind of response goes down as people get older. Reduced adaptive immune responses can also occur. We have less of a repertoire of T cells that can recognize foreign antigens, and so we're at greater susceptibility. And there's reduced blood-brain barrier function too with older age that can probably link to increased inflammation, and so viruses may have a better segue to get into the central nervous system and cause problems. All right, the last one is Zika virus. So Zika, again, Flaviviridae. It's spread by Aedes aegypti mosquitoes that are like to snack on us in the daytime. They carry the virus, and the virus then replicates in the mosquito midgut, the epithelial cells, and the salivary glands. So when it injects it to take a meal, it leaves behind a present. And once it's inoculated in the skin, the virus can infect various cell types like keratinocytes and so on. It can spread then through the lymph nodes to the bloodstream. And what we really worry about with Zika is the fact that it can cause very harmful fetal microencephaly. So adults who get Zika are not getting, you're not going to get sick. You might have a little headache or something, but the symptoms are mild, and it's nothing. But it is very serious for someone who's pregnant who gets infected, and then the fetus may develop poorly. And this is a major issue, and has been a major issue. If you look back historically at Zika, we've known about it since the 1940s. Monkeys and mosquitoes in Uganda were spreading the virus around. In the 90s, it was circulating in West Africa. And in 2007, the first reported outbreak outside of Asia and Africa occurred. And then in 2015, it spread to the Americas. And this is when it really started causing all these microencephaly problems. which really took off at this time in 2015. The World Health Organization in 2016 declared Zika was no longer a global emergency, although it had been declared an emergency right prior to that. This is because Zika infections have fallen dramatically over the years. You can see from the map that our friends in Central and South America have had a lot of problems with Zika viruses, but it's right on our doorstep. But the good news is a lot of infections have gone down since that time. And the most likely explanation is thought to be herd immunity, that a lot of people have become resistant. They've been exposed, they weren't pregnant or whatever, and so now the virus is not spreading as much in the resistant population. In the United States, we have had cases of Zika since 2015 through 2018. 5,653 symptomatic Zika cases have been described. Most of them, the vast majority, are travelers returning or coming from an endemic area. So there are some cases where people think that local mosquitoes have contributed. This is in Florida and Texas, and so that's something to be concerned about. And then there's the possibility of sexual transmission too that has been recognized in several cases. Zika is a really good example of how a viral mutation can affect the outcome of a disease. In this case, there was a single mutation. If you go back to when Zika started spreading to the Americas, what you find is a specific mutation in the PRM protein that seems to confer an ability of the virus to infect the central nervous system much better. And so in these studies, this was done with mice, and they were injecting the virus into the central nervous system. And they were using isolates from 2010, 2015, and 2016 from different areas. And what they found is that the 2010 virus that they injected into the animals did not cause much death. It was only around 15–20% of the animals would die when they gave them that virus. But if they gave them a more recent isolate from 2015 and 2016, the animals died much more quickly. They all died. So that's what that plot on the left shows. And then on the right, it just shows you some changes that occur in the brain tissue itself as a consequence of infection with the mutant versus the virus. So a mutation is responsible then for the more severe infection. So what's a way to strategize and prevent Zika? Well, we do have some very promising vaccines actually out there that have been protective in animals and looking pretty good. But the problem is we can't test them in humans because the infection rates have dropped so much that it's hard to it's hard to find a population of people who are susceptible that you can actually take the vaccine in the field and show that there's efficacy. So that's a problem for the development of the vaccine at this point. Maybe that's a good problem. I guess it is. But there are some other ideas out there, and people are using methods to genetically engineer mosquitoes that are resistant to the virus itself. And so this is being done for Zika, for dengue virus, and some others. The idea here is that you basically make a mosquito that has genetic resistance to the virus. And the virus cannot replicate in the mosquito. And they use these synthetic small RNA molecules. So small RNA molecules, that's one of they're regulatory in their effects. They can bind to messenger RNA, and they can prevent messenger RNA from replicating or from being translated. And so what they do is they make these mosquitoes so that they make these small RNA molecules. And the small RNA molecules then are going to block the virus from replicating in the mosquito. They're going to bind to the transcripts of the virus in the mosquito and prevent replication there. And so now the mosquito can't transmit. And then they put in this really cool fluorescent protein into the eyes of their mosquitoes so that they can shine a fluorescent light on them and tell if they're if the mosquito they're looking at is the one that they've genetically engineered or some others. And this is sort of the idea, well, when we release, if and when they release these into the wild, they'll be able to tell if their mosquito is doing okay population-wise and so on. Distinguish it from the others with a relatively easy method. And so far this is just done experimentally. So they took mice and they took Zika virus, they put Zika virus in a bag of blood, if you will, and they let the mosquitoes chow down on it. So the mosquitoes had a blood meal with either, and they used either wild type mosquitoes or they used mosquitoes that had these genetically manipulations, so they would be resistant to the virus. And then they let the mosquitoes chow down on mice. And what they find is that if they use wild type mice, these Higgs, or wild type mosquitoes rather, these Higgs guys, that the mosquitoes were just fine at transmitting the virus. It grew platforming units, PFU, that's how much virus is being made in the animal, lots of virus. But if they use the anti-Zika, and a couple different types of anti-Zika mosquitoes, they were able to knock down that production of virus in the mosquitoes themselves. And then when they are, I'm sorry, that was in the mosquito, the viral replication. And then when they let the mosquitoes chow down on the mice and say, okay, we're going to use STAT1 knockout mice. All right, so these are mice that don't have a good immune response to type 1 interferons. They can't generate that sort of branch of the innate immunity because that's a downstream transcriptional factor that is induced by type 1 interferons. So these are very susceptible mice. And if they infect these mice with wild type virus, the mice start losing weight. Whereas if they use the mosquitoes that have the anti-Zika properties, they don't lose weight, they look healthy, and then they survive, or they don't survive based on that. So this is pretty exciting stuff, kind of interesting, little science fiction type thing that you wonder if it's a good idea to release genetically engineered insects into the environment, but it is sort of clever. All right, so that does it for RNA Viruses 1. All right, I got you half hour.
Thank you.
Thank you.
Good luck, we'll see you tomorrow.